Hey everyone, in this last video for chapter 7, we're going to be looking at bulk transport across the plasma membrane. Bulk transport occurs in two different ways, through exocytosis and endocytosis. Small molecules in water can enter and leave the cell through the lipid bilayer via transport proteins. Large molecules such as polysaccharides and proteins have to cross the membrane and bulk inside of vesicles. So, bulk transport is the way you can transport large things across the plasma membrane. Uh, so, in exocytosis, things are exiting the cell. So, you remember EX is exiting. So, exocytosis, transport vesicles can migrate to the membrane, fuse with it, and release the contents outside of the cell. This happens constantly, all the time. Some cells are programmed to do certain functions or jobs, or they're more focused on creating certain uh, types of products that need to be secreted out of the cell. And so they need to do a lot of exocytosis. I love the example in that the pancreas will secrete insulin um, by exocytosis. And so that's just one of many examples of how exocytosis is used um, by the cells. So the Golgi is, remember, is the shipping and warehouse of the cell. The Golgi will create vesicles and package the contents that are needed to be sent outside of the cell. So the vesicle will bud off of the Golgi and it will travel towards the um, plasma membrane, where it will fuse with the membrane and release the proteins or items that it's needed to be released. That vesicle can then be used as becomes part of the plasma membrane. And remember, so when things start to enter the cell, parts of the plasma membrane will be converted back towards vesicles. And so for things to enter the cell, that's called endocytosis. Endo meaning to enter, enter. Endocytosis is things going into the cell. Now, endocytosis has different kinds of different ways of doing this. So the membrane will form a pocket around whatever it is it's trying to take in, forming a vesicle, and that will travel into the cell. There are three types of endocytosis. There's phagocytosis, pinocytosis, and receptor-mediated endocytosis. Phagocytosis is cell eating. Pinocytosis is commonly called cell drinking. And so let's go over these in image form. So here's phagocytosis on the left. Phagocytosis is cell eating, in that um, the cell will form a extend around the object it's trying to pull in and form a vesicle, and it will pull it inside of the cell to be used. So the next step in this process is either a lysosome or something will come along and digest um, the food and convert it into the molecules that are needed for the cell to do its job. Pinocytosis is cell drinking, in other words, which it could be water molecules if it needs water, bulk water transport, but usually it's some sort of fluid that contains the solutes. And so pinocytosis is commonly called cell drinking. And so you can see um, a coated a, the membrane can have a coated protein to indicate that it's searching for a certain specific kind of solute. And so it will do that to try to pull in as many solids as possible. Receptor mediated endocytosis is basically targeted endocytosis, in that you have receptor proteins that are searching for or targeting a specific solute. And then, as enough of them are bound or start to be utilized, the vesicles will pull in those receptors and take them into the cell to be used for whatever purpose that they may be needed. Um, in phagocytosis, it's also the cell will basically extend around the object, and so a great example of that would be like your white blood cells in your body. An amoeba will engulf a green algal cell via phagocytosis. It will completely surround the algal cell and take it in and use it as food. This is just showing you how pinocytosis is. Pinocytosis is actually harder to see um, under the microscope because you're not really seeing a whole lot, but you can see uh, you're not seeing a solid object like entering the cell, but you will see the vesicles start to uh, bubble inwards for the cell. And in receptor mediated endocytosis, the example here, you can see how you will have a plasma membrane with coat proteins that are searching, receptor proteins that are searching for a specific item. And then once it gets enough of those, it will bud inwards, creating a vesicle. So human cells use receptor mediated endocytosis to take in cholesterol, which is carried by particles called LDLs, low density lipoproteins, and then individuals with familial hypercholesterolemia have missing or defective LDL receptor proteins. So cholesterol accumulates in the blood, building up lipids and narrowing the space in the blood vessels, resulting in potential heart damage or stroke. This is usually a genetic defect that they may have. And so people who have this have to take you have to take medicine and cholesterol drugs to maintain the LDLs. You guys may have know of like if you ever go to the doctor to get your cholesterol check, they'll tell you you have either too many LDLs and not enough HDLs. It just lets you know which if you need to be eating more healthier fats or not in your diet. And so with that, guys, that concludes chapter 7 about the plasma membrane.